Let's resume. Good afternoon, everybody. Let's resume. Now, before going into the break, we discussed cloning, reproductive cloning, and also what is called as therapeutic cloning. Now we have to discuss another topic, and this is called as what? That's called as triple parent baby. So let's see that what is the meaning of triple parent baby. Triple parent baby. So what is the meaning of triple parent baby and how this baby would be developed? See, first of all, when fertilization takes place, suppose this here, the fertilization is taking place. This is an egg and this consists of the nucleus. This is the nucleus and here is mitochondria of the egg. So when fertilization takes place, what happens? That the sperm of father is responsible for fertilizing the egg of the mother. Now, the genetic material, when fertilization is taking place, the genetic material of father is reaching the egg. But the genetic material would be confined to the nucleus of the egg. Here I am saying that when we talk about fertilization, we can see when fertilization occurs, the genetic material of the father would be reaching only and only the nucleus of the egg. And where the mother's genetic material is also present. So I have written F and M, father's genetic material and mother's genetic material. Clear is, so whenever fertilization is taking place, the genetic material of the father is reaching only the nucleus of the egg. Clear? It does not reach the mitochondria. So it means what? That the mitochondrial DNA which is present in us belongs to our mother, not to our father. So the cells that we have, in those cells, how can the donor and the egg provider be the same? The donor and the egg provider would be the same because somatic cell would be taken from the same lady and the egg would be taken from the same lady. Clear? So from her egg, the nucleus would be removed. And if suppose you ask why the nucleus is of her only, why should it be removed? Then the reason is that egg cell is what? Egg cell is a reproductive cell. It has half the number of chromosomes. It cannot develop into a baby. And somatic cell of the same lady would be having the proper set of chromosomes, 23 pairs, which would be developing into a baby. Definitely. Clear? So that's why the egg provider and the donor would be the same lady as such, the same person as such. In that case, the mitochondrial DNA belongs to the same lady. Mitochondrial DNA would be belonging to the same lady because it has not been removed. Clear? In case of the reproductive clone that we have discussed, the donor was suppose the male and the female was the egg provider. So the mitochondrial DNA would be belonging to the egg provider, not the donor. So how can you say that the genetic material of the donor is same as the genetic material of the clone, because there's a difference between the two. Clear? So that's the reason why we are saying. Now here we can see that the egg is being fertilized by the sperm and the genetic material of the father reaches only the egg. It hardly reaches the mitochondria. Clear? So mitochondria that we have in our body belongs exclusively to our mother, not to our father as such. Clear? Now what can happen? That as far as mitochondrial DNA is concerned, out of 20,000 genes that we have in our cells, 37 genes are determined by mitochondrial DNA. 37 genes are determined by mitochondrial DNA. And these would be responsible for some problems. Clear? 37. Sometimes they can be, these mitochondrial DNA may be responsible. Generally they are not. But sometimes they would be associated with disorders. And these disorders would be directly passing from the mother to the child. These disorders would be directly passing from the mother to the child. Clear? So it would be transferred from the mother to the child. Fine, these disorders. And there would be a big problem in this. And if suppose a lady knows that, because the lady would be also suffering from this disorder. She is having a dysfunctional mitochondrial DNA in her body. And if she reproduces, then the same disorder is being transferred from the mother to the child. I'm saying what? That in this case, the mitochondrial DNA in me also, mitochondrial DNA belongs to my mother. In you also, mitochondrial DNA belongs to your mother. So if the mitochondrial DNA is dysfunctional, is responsible for diseases, is associated with diseases, what will happen? The disease would be transferred from the mother to the child. Clear? The disease is getting transferred from the mother to the child. Now, if a lady knows that her disease is going to be transferred from her to her own child, she won't undergo pregnancy, but she has a right to have a baby. And for this purpose, triple parent baby is developed. Clear? So for this purpose, triple parent baby is developed. So first of all, what we are responsible for doing, we are responsible for, this is suppose dysfunctional mitochondria. This we say is dysfunctional mitochondria in this fertilization. So we are responsible for we are responsible for removing this mitochondria. So it would be removed. Not mitochondria would be removed. First of all, mitochondria cannot be removed. It's called as the powerhouse of the cell. So let's see how the transfer of mitochondria takes place. We are responsible for removing the nucleus. And this nucleus is consisting of what? The genetic material of father and mother. Clear? Now we'll select another mom. We'll select another mom. Another lady. And her egg would be extracted. So another lady. Now her egg would be, first of all, it would be examined that whether her mitochondrial DNA is proper or not, whether it is responsible for any disease or not. So first of all, the egg would be extracted from her. Egg would be extracted from her. And here, her nucleus, here, this is mitochondria of hers. Let's name it M1. Clear? So the nucleus of the lady would be removed. So we are responsible for removing the nucleus. Clear? And in the place of this, the nucleus of the fertilized egg would be transferred. So we are responsible for transferring the nucleus of the fertilized egg here. So this is being removed and this is being transferred. Clear? Now finally, in the egg we have the combination what? That in the nucleus, the genetic material of the father and the first mother is present. And in the mitochondria, the genetic material of the second mother is present. This is how mitochondrial DNA transfer can take place. You cannot remove mitochondria from the egg because it is the powerhouse of the cell. The cell would be destroyed. You cannot do that. So this is how mitochondrial DNA would be removed and transferred. So we can see that this would be the final combination. Now this would be planted in the womb of, this would be transferred to the womb of surrogate mother. Surrogate mother. The baby that is born, would be free from mitochondrial DNA disorder. Why? Because the mitochondria has been the second mother's mitochondria is present in her, and the first mother's genetic material is present in the nucleus. The father's genetic material is present in the. So she would be free from mitochondrial DNA disorder. She would be free from mitochondrial DNA disorder. But the problem is that she would be having one father and two mothers. Clear? She would be having one father and two mothers. And that is what is called as triple parent baby. So the womb can be that of the first mother, second mother, or surrogate mother. But remember that this is not surrogacy. The second mother has a genetic connection with the child because her mitochondrial DNA is present in the child. So some countries like UK, Canada, the Australia, they have given a go ahead for development of this kind of baby. They have given a go ahead for development of this kind of baby. But if we are responsible for approving it, clear, in India, we should sort out the legal concern because we should know that under what condition the second mother would be laying a claim on the baby. So this is called as triple parent baby. Fine. So you have to know this is based on mitochondrial DNA transfer around triple parent baby. Triple parent baby. Parent baby. Triple parent baby. Parent baby. Write down. If cloning is based on somatic cell nucleus transfer, if cloning is based on somatic cell nucleus transfer, somatic cell nucleus transfer, if cloning is based on somatic cell nucleus transfer, triple parent baby, triple parent baby, parent baby is based on, triple parent baby is based on mitochondrial DNA transfer, is based on mitochondrial DNA transfer. Triple parent baby is based on mitochondrial DNA transfer. Mitochondrial DNA transfer. Mitochondrial DNA transfer. Clear? And as far as triple parent baby is concerned, do remember that out of 20,000 genes that we have, 37 genes are determined by mitochondrial DNA. Right? Out of 20,000 genes that we have, out of 20,000 genes that we have, 37 genes out of 20,000 genes that we have, 37 genes are determined by mitochondrial DNA, are determined by mitochondrial DNA. And we can see that this is directly transferred, mitochondrial DNA is directly transferred from the mother to the child. No role is played by father in it. Write down mitochondrial DNA is directly transferred from the mother to the child. Mitochondrial DNA is directly transferred from the mother to the child. Directly transferred from the mother to the child. Mother to the child. No role is played by father in it. No role is played by father in it. Mother to the child. No role is played by father in it. Played by father in it. Clear? Father in it. Sometimes mitochondrial DNA. Sometimes mitochondrial DNA is responsible for disorder. Sometimes mitochondrial DNA is responsible for disorder. Responsible for disorder. Mitochondrial DNA is responsible for disorder. Disorder which would be transferred directly from the mother to the child. Which would be transferred directly from the mother to the child. From the mother to the child. Mother to the child. And we can see, in order to remove this drawback, triple parent baby is used. Right? Now, in order to remove this drawback, in order to remove this drawback, triple parent baby is used. Triple parent baby would be developed. In order to remove this disorder, triple parent baby would be developed. First of all, triple parent baby would be developed. First of all, the nucleus of the fertilized egg is removed. First of all, the nucleus of the fertilized egg is removed. And since this now consists of, after the removal of nucleus, this would be consisting of the dysfunctional mitochondria. So the cell would be discarded. First of all, the nucleus of the fertilized egg would be removed and the cell would be discarded. Cell would be discarded. Clear? So that would be done. Discarded. Thrown away. Another mother would be selected. With proper mitochondrial DNA. Another mother would be selected with proper mitochondrial DNA. Her egg would be removed. And the nucleus of fertilized egg would be transferred. Her egg would be removed and the nucleus of the fertilized egg would be transferred to it. Nucleus of the fertilized egg would be transferred to it. And Clear? Now, nucleus of the fertilized egg would be transferred to it. Now, this would be planted inside the womb of surrogate mother. Clear? And the baby that is born would be free from mitochondrial DNA disorder. Write down. It would be planted in the womb of surrogate mother. The baby that is born would be free from mitochondrial DNA disorder. The baby that is born would be free from mitochondrial DNA disorder. But would be having one father and two mothers. Clear? So this is called as triple parent baby. And do remember that in India, in this case, the second mother has a genetic connection. It's not surrogacy as such. Surrogate mother we have used, but surrogate mother in India would be direct relatives only, according to the Supreme Court. So the surrogacy, surrogate mother in India would be only and only direct relatives only, no other person. Surrogate mother. Clear? Direct relatives only. No other person can be surrogate mother in India. That has been passed by the Supreme Court, because in India surrogacy was done on a large scale, and when foreign couples used to come, surrogate mother were selected from India. When the time of separation came, they desisted from separate from the child. So it resulted in a number of cases in India. That's why the Supreme Court said that surrogate mother can be only and only direct relatives. No other can be surrogate mother. Now, that's your triple parent baby. Now, whatever we have discussed is nothing but biotechnology. But as far as biotechnology is concerned, we can see that biotechnology would be having two broad forms. One form is called as tissue culture. So if we talk about biotechnology, there are two broad forms of biotechnology. One is called as tissue culture, and second is called as what? Second is called as genetic engineering. Genetic engineering. Tissue culture, and second is called as genetic engineering. Tissue culture means what? Tissue culture means that this culture, when we say first of all culture is the growth of cells of plants and animals on a large scale. Growth of cells of plants, animals, and microorganisms also on a large scale in a controlled condition in the lab. So that's called as culture. Tissue culture means it is the growth of cells of plants and animals barring microorganism. It's the growth of cells of plants and animals, and it does not include microorganism. So that's called as what? That's called as tissue culture. That is called as cell culture. So it would be called as tissue culture, or it can be called as cell culture also. Clear? Cell culture or tissue culture. Now, as far as it is concerned, when you're talking about tissue culture, we can see that for this purpose, what we are doing, we are selecting a medium, and this medium can be semi-solid or liquid. So we sterilize that medium. Sterilize that medium. Clear? And then we are responsible for adding, sterilize means germ-free, and then we are responsible for adding hormones and nutrients to the medium, so that the multiplication of cells takes place on a large scale. We would be also responsible for changing the medium. The nature of medium won't be changed. The same medium would be used, but nature of medium won't be changed. The same medium is used, so we are responsible for changing the medium also. Clear? Changing the medium also. And when we change the medium, we can see that we are responsible for changing. The same medium would be used, but the changing of medium is mainly for the purpose of what? Changing of medium is mainly for the purpose of removing what? Removing the waste products which has developed in this process. Clear? Now we can see that also that as far as tissue culture is concerned, as far as tissue culture is concerned, uh, tissue culture was successfully used in India by the National Chemical Lab of Pune. And it was used for what purpose? It was used for developing varieties of bamboos which can flower in weeks instead of years. Bamboo flowers in years. When it flowers, it dies. So there was a difficulty in developing the hybrid varieties of bamboos. There was a difficulty in developing the hybrid varieties of bamboos. Clear? So with the help of tissue culture, in the National Chemical Lab of Pune, bamboos varieties were developed which can now flower in weeks instead of years. So this was a big accomplishment. Now, as far as genetic engineering is concerned, genetic engineering means what? That after the unveiling of double helix structure by Watson and Crick, genetic makeup of an organism can be changed. Now, genetic makeup of an organism can be changed how? By either the deleting an existing gene from it, or by introducing a new gene into it. So the genetic makeup can be changed. And genetic engineering is also called as recombinant gene technology. It is also called as genetic manipulation. In case of tissue culture, there is no genetic manipulation. And there is vast application of genetic engineering in many fields. For example, agriculture, to develop GM crops. For example, inbuilt insecticide in plants. For biofortification. So vast application. Application is in the field of medicine also. Application is in field of agriculture also. So many areas the application can be seen. Clear? Right on. Biotechnology has two broad forms. Biotechnology has two broad forms. First, tissue culture. First, tissue culture, and second, genetic engineering. First, tissue culture, and second, genetic engineering. Genetic engineering. Two broad forms, tissue culture, and second is genetic engineering. Engineering. Tissue culture is the growth of, tissue culture is the growth of cells of plants and animals. Growth of cells of plants and animals. Cells of plants and animals. On a large scale. On a large scale. Cells of plants and animals on a large scale in a controlled condition. In a controlled condition. Controlled condition in the lab. In a controlled condition in the lab. Controlled condition in the lab. When exclusively, in a controlled condition in the lab. That's called as tissue culture. We are not including within tissue culture microorganism. In culture, microorganism is included, but in tissue culture only plants and animals. For this purpose, a medium is selected. For this purpose, a medium is selected. Medium is selected, which is either liquid or semi-solid. Medium is selected, which is either liquid or semi-solid. Or semi-solid. Liquid or semi-solid. The medium is sterilized also. The medium is sterilized also. Sterilized means germ-free. The medium is sterilized also, germ-free. Hormones and nutrients. Hormones and nutrients. Hormones and nutrients are added to the medium. Hormones and nutrients are added to the medium. Nutrients are added to the medium. So that multiplication of cells takes place. So that multiplication of cells takes place. Added to the medium. So that multiplication of cells takes place. Multiplication of cells takes place. Clear? Multiplication of cells takes place. And the medium would be constantly changed also to remove the waste product. Write down. The medium is also constantly changed. The medium is also constantly changed. The medium is also constantly changed to remove the waste product. The medium is also constantly changed to remove the waste product. Constantly changed to remove the waste product. The medium is also constantly changed to remove the waste product. But do remember that there is no genetic manipulation, no genetic alteration in tissue culture. Write down. There is no genetic alteration, genetic manipulation. There is no genetic manipulation in tissue culture. There is no genetic manipulation. Genetic manipulation in tissue culture. In tissue culture. There is no genetic manipulation in tissue culture. In tissue culture. Clear? No genetic manipulation takes place in case of tissue culture. And one thing can be written is the biggest accomplishment of National Chemical Lab of Pune. Write down. The National Chemical Lab of Pune. The National Chemical Lab of Pune. The National Chemical Lab of Pune. Lab of Pune. With the help of tissue culture. With the help of tissue culture. Tissue culture has developed varieties of bamboos. Has developed varieties of bamboos. varieties of bamboos which can flower in weeks instead of years, which can flower in weeks, which can flower in weeks instead of years, which can flower in weeks instead of years, instead of years. So that accomplishment is of National Chemical Lab of Pune. Clear? So that's about tissue culture. But when we talk about genetic engineering means what? It means that an exotic, write down genetic engineering, discover of genetic engineering, and then we deal with what is gene therapy, what is CRISPR-Cas9 technology, designer baby. Fine, genetic engineering. Genetic engineering. It is also referred to as. It is also referred to as recombinant gene technology. It is also referred to as recombinant gene technology. Recombinant gene technology or genetic manipulation. Recombinant gene technology or genetic manipulation or genetic manipulation. Recombinant gene technology or genetic manipulation. Clear? Fine. Genetic manipulation. So that's called as genetic engineering. Genetic manipulation or recombinant, same thing. So what happened? After the double helix structure of DNA was given, it became possible to change the genetic makeup of organisms. Right on, after the double helix structure was given, after the double helix structure was given, the double helix structure was given, helix structure was given, it became possible, it became possible to change, it became possible to change the genetic makeup of organisms. It became possible to change the genetic makeup of organisms. Genetic makeup of organisms by either introducing a new gene into it, by either introducing a new gene into it, a new gene into it, gene into it, or by changing, or by changing an existing gene, or by changing an existing gene, changing an existing gene, changing an existing gene. Clear? Existing gene. Clear? So this is called as genetic manipulation, or the resultant gene would be called as what? It would be called as recombinant gene. Write down. The resultant gene is called as recombinant gene. The resultant gene is called as recombinant gene. Recombinant gene. Is that clear? So that resultant gene would be referred to as recombinant gene. So this is genetic engineering. Now, it has vast application in the field of agriculture and also in the field of medicine. Write down. It has vast application in the field of agriculture. It has vast application in the field of agriculture. Application in the field of agriculture. And also in the field of medicine. And also in the field of medicine. Also in the field of medicine. See, when you talk about some technologies here need to be discussed. One technology is called as what? One is called as, not technology, in fact it's therapy. One therapy is called as gene therapy. Gene therapy. Now, gene therapy was developed by, we can see, now also in biotechnology, you have to know gene therapy, CRISPR-Cas9 technology, on which questions are drafted, and also GM crops also. So let's cover up all these topics so that we can complete biotechnology. See, gene therapy means what? Gene therapy means that it is a mechanism under which, it's a therapy under which inherited disorder in a person can be removed. Inherited disorder means the disorder is being passed from one generation to another generation. So inherited disorder in a person would be removed. So that's called as gene therapy. But how it is conducted? This method was given by W. French Ederson. Method was developed by W. French Ederson. And as far as W. French Ederson is concerned, he developed this method. Now, first of all, what we do is that we are responsible for identifying the faulty gene. See, it is an inherited disorder. It means that a faulty gene would be responsible for it. So we identify the faulty gene which is responsible for the disorder. And in the lab, we are responsible for making the normal version of that gene. And we make the normal version of that gene by the process of cloning. Cloning means somatic cell nucleus transfer. Clear? So the gene would be extracted from the nucleus. So we are responsible for making the normal version of that gene through cloning. We are not developing the gene as such, but we extract it through cloning and then it would be transferred. Now, when you are responsible for developing normal version of the gene, you now have to transfer this gene to the cell. Then only the cell would be, the disorder in the cell would be resurrected. So what you do is that you develop the normal version of the gene by cloning, and then after, what you are responsible for doing, you are responsible for suppose you have developed the normal version. Now what you do is you select some body, select the cells from the body of the patient. You get some cells, not select. You are responsible for extracting some cells from the body of the patient. And then the normal version genes would be transferred into it. So when you are responsible for transferring the normal version genes in the somatic cells or the cells of the patient, then the disorder in these cells would be resurrected. Then these cells would be called as doctored cells. Doctored cells. These cells are called as host cells, which are selected from the body of the patient. Now, these doctored cells would be returned back to the body of the patient through injection. Through injection. Just 1 second. Just 1 second. Just 1 second. Just About uh, gene therapy. See, you have asked me a question. Please explain about the lab grown meat. See, lab grown meat would be grown in two ways. Lab grown meat is what? Moza meat that we have discussed earlier. And moza meat is grown in the lab by taking a thin line of cells from these animals whose meat we are considering. And then we are responsible for using tissue culture to develop that meat. So the claim of that is what? The claim of that is that the taste of the meat is the same, the nutrient value is the same. So this would be available in the market within a period of 3 or 4 years. That has already been developed. So commercial for sale, it would be available in the market. But as far as lab grown diamond is concerned, lab grown diamond is nothing but a cultured diamond. Through tissue culture, it has been developed. Clear? So all these are mainly developed through tissue culture. And the purpose of developing this meat is what? That if suppose we consume the meat, natural meat, you have to grow these animals in large numbers, ruminants. And these animals are big contributors in global warming because they emit methane, nitrous oxide, and CO2. And you have discussed that 14% of global greenhouse gases emission takes place by these animals only. Clear? So that's the reason why we are talking about it. Now, this would be these doctor cells would be injected into the body of the patient. Now, when these cells replicate inside the body, then the disorder would be gradually removed. But there's a problem in this. There's a problem in this. The problem is what? The problem is that when again this returns back, see what happens if suppose I am the patient and gene therapy is conducted on me, I would be cured. But in my next generation, subsequent generation, the disorder would again return back. Why? Because gene therapy is only conducted for somatic cell. It is never conducted for reproductive cell. So the reproductive cells are responsible for carrying the disorder to subsequent generation. And again in the subsequent generation, gene therapy would be conducted. Clear? Gene therapy would be conducted. Write down gene therapy. Gene therapy. Gene therapy. It was developed by, it was, this therapy was developed by gene therapy. This therapy was developed by W. French Ederson. This therapy was developed by W. French Anderson. W. French Anderson. This therapy was developed by W. French Ederson. W. French Ederson. Was developed by W. French Ederson. Clear? W. French Ederson. Fine. And we can see that this is for removing inherited disorder which would be seen in a person. Write down. This is for the purpose of. This is for the purpose of removing. This is for the purpose of removing inherited disorder. This is for the purpose of removing inherited disorder. Inherited disorder. Disorder in a person. This is for the purpose of removing inherited disorder in a person. Inherited disorder in a person. Clear? In a person. Now, inherited disorder means that it is transferred from one generation to another. Write down. First of all, the faulty gene responsible for the disorder. First of all, the faulty gene responsible for the disorder. Faulty gene responsible for the disorder is identified. The faulty gene responsible for the disorder is identified. And faulty gene responsible for the disorder is identified, and normal version of this gene, normal version of this gene, normal version of this gene is prepared in the lab. Normal version of this gene is prepared in the lab by cloning. Cloning means somatic cell nucleus transfer. So the normal gene would be extracted from that nucleus, and the normal version of this gene is prepared in the lab through cloning. Through cloning. Clear? So that would be done. And now what you have to do is, now you have to uh, transfer that gene. So some host cells would be selected from the body of the patient. Right on. Host cells are selected from the body of the patient. Host cells are selected from the body of the patient. Selected from the body of the patient. And normal version genes, host cells, are selected from the body of the patient, and normal version genes are transferred into it, and normal version genes are transferred into it, Now, when you are transferring the normal version gene in the host cells, these cells would be called as what? These cells now would be referred to as doctored cells. Write down. These cells are now referred to as, doctored cells. These cells are now referred to as doctored cells. Which is returned back to the body of the patient. Returned back to the body of the patient. Through injection. Returned back to the body of the patient through injection. Returned back to the body of the patient through injection. When these cells replicate, they start to replicate. The disorder would be gradually removed. When these cells replicate, the disorder would be gradually removed. Clear? The disorder would be gradually removed. But then the problem is there, that the disorder would be again returning back in the next generation. Right on. But the disorder returns back. In the next generation. In the next generation. And why the disorder would be returning back in the next generation? Because gene therapy is only conducted for somatic cell. It is never conducted for reproductive cell. Right on. As gene therapy is only conducted for somatic cells. And never for reproductive cells. Clear? That's the reason why we won't be able to remove the disease in the subsequent generation, because for reproductive cells there is no gene therapy as such. Clear? No gene therapy as such. Fine. Now, if suppose you have to indulge in genetic alteration in reproductive cells or embryonic cells. Clear? Reproductive cells or embryonic cells. It would be done also for somatic cells. So if suppose one has to indulge in genetic alteration in reproductive cells or embryonic cells, then we can see that we would be using what? We would be using a technique which is called as what? Which is called as CRISPR-Cas9 technology. Clear? CRISPR-Cas, and on CRISPR-Cas9 technology, they ask you questions. CRISPR-Cas9 technology, they ask you questions. So let's see what is CRISPR-Cas9 technology. See, CRISPR, sorry, CRISPR-Cas9 technology. Now, what is CRISPR-Cas9 technology? CRISPR stands for what? It stands for clustered. R for regularly. I for interspaced. IS for interspaced. Palindromic. Repeats. Palindromic repeats. And Cas9 is what? Cas9 is a type of enzyme. Cas9 is a type of enzyme. Clear? So clustered, regularly interspaced, palindromic repeats, and Cas9 is what? Cas9 is a type of enzyme. Clear? So that's called as CRISPR-Cas9 technology. But what is this? Now this technology would be used. We know that drawback of gene therapy is what? That it cannot be utilized for reproductive cells. So for genetic alteration in reproductive cells, for genetic alteration in embryos, for genetic alteration in also embryos, reproductive cells, as well as somatic cells also it can be used. We use what? We use CRISPR-Cas9 technology. Now this technology was developed by Doudna and Carpentier. They were given the Nobel Prize also in the field, Nobel Prize in the year 2020 for this purpose. And it's not that all the diseases would be cured. All the diseases would be cured by gene therapy. It's not that. Only some diseases can be cured. Now, when you talk about thalassemia, it cannot be cured by gene therapy. Clear? It's not that. I have told you the restriction of gene therapy is what? There is a restriction of gene therapy. So it's not that all diseases can be cured. Only few diseases which are inherited would be cured. And that too also, do remember that it can be done only in somatic cell, it cannot be done in reproductive cells. So that's the restriction of gene therapy. But when we are talking about CRISPR-Cas9 technology, this technology can be used for removing, or this technology can be used for genetic alteration in not only somatic cell but also in reproductive cell, that is called as germline cells, or also embryos. For that purpose also it can be used. Reproductive cells, embryos, for that purpose also it can be used. So that is CRISPR-Cas9 technology. But how and what is CRISPR-Cas9 technology? See, as far as CRISPR-Cas9 technology is concerned, CRISPR-Cas9 technology has two important tools. Now, the first tool of CRISPR-Cas9 technology is what? It is a segment of two important tools are present in CRISPR-Cas9 technology. First important is what? Suppose this is the genetic makeup which needs to be altered. This is the genetic material which needs to be altered. And this is the DNA segment that you have to remove. So CRISPR-Cas9 technology consists of what? It consists of a segment of RNA. Now this segment of RNA is first part of CRISPR-Cas9 technology, which would be binding with the segment of DNA which you need to remove. So that's a segment of RNA which is going to bind with segment of DNA which needs to be removed. And then we would be responsible for using Cas9. So Cas9 is what? Cas9 is a molecular seizure. molecular scissor, is a molecular scissor which would be responsible for striping off, removing it, acts as a molecular scissor which would be responsible for removing this segment of DNA. And on the place of that, a new segment can be introduced. On the place of that, a new segment of DNA can be introduced. So that's how it is work. I'm saying that there are two tools in CRISPR-Cas9 technology. First tool is what? A segment of DNA. This segment of DNA is going to bind with a segment of RNA which is going to bind with segment of DNA which needs to be removed. And then we have Cas9 is nothing but a type of enzyme. So Cas9 is acting as what? It is acting as a molecular scissor. Now this would be responsible for stripping off the segment of DNA which needs to be removed. Stripping off the segment of DNA which needs to be removed. Clear? So that's how CRISPR-Cas9 would be working. Now, with the help of CRISPR-Cas9 technology, we can develop designer babies. Designer baby means that this kind of baby would be fulfilling the aspiration of parents. Parents wants these babies to be. CRISPR-Cas9 technology has, first of all, a positive impact. Positive impact means that this technology was used to develop a number of hybrid varieties which enhances agricultural production. This technology was used for, it has wide range application in the field of agriculture. Inbuilt insecticides, etc. can be developed. So it enhanced crop production, it removed diseases associated with the crops. And for this purpose, for this contribution of CRISPR-Cas9 technology, the scientists who were responsible for developing it, called Doudna and Carpentier, they were conferred the Nobel Prize in the year 2020. But see, CRISPR-Cas9 technology can be criticized also. If CRISPR-Cas9 technology is used for removing diseases, then it is wise. It can be used. But if CRISPR-Cas9 technology is used for fulfilling the aspiration of parents, that the parents wish that these kind of features should be present in their child, then it is not justified. It is not justified. In this context, we can talk about the development of designer babies. On the other hand, when we are talking about the CRISPR-Cas9 technology, we can see that CRISPR-Cas9 technology can be used for other purposes also. Not only has been used for this purpose, it can be used for other purpose also. For example, Jian Kui, a Chinese scientist, he was responsible for developing twins with the help of CRISPR-Cas9 technology who have innate resistance to HIV. Inbuilt resistance to HIV. And how he did? By developing a mutated version of a gene which is called SCCR5 delta 32. So that disallows the invasion of HIV inside the body. So this was a contribution made by whom? Made by Jian Kui. Clear? So all these comes under the CRISPR-Cas9 technology. But CRISPR-Cas9 technology can be criticized also, that if suppose you are responsible for doing any kind of alteration in reproductive cells, it can pass on to the subsequent generation. You have no answers to it also. Let's include all these in our discussion. Write down CRISPR-Cas9 technology. CRISPR-Cas9 technology. Clustered, regularly interspaced, palindromic repeats. Clustered, regularly interspersed palindromic repeats. Palindromic repeats. That's the full form of CRISPR. CRISPR-Cas9 technology. This technology was developed by Dodna, Dodna, and Carpentier. This technology was developed by Dodna and Carpentier. Dodna and Carpentier. Technology was developed by Dodna and Carpentier. This technology has two tools. This technology has two tools. Two tools. Two tools. A segment of RNA, a segment of RNA, segment of RNA, which binds with the segment of DNA, which binds with the segment of DNA that needs to be removed, which binds with the segment of DNA that needs to be removed, that needs to be removed, segment of DNA that needs to be removed, and second, second segment of DNA which that needs to be removed, and second, Cas9, Cas9, which is a type of enzyme, Cas9, which is a type of enzyme, and acts as molecular seizure. Cas9, which is a type of enzyme and acts as a molecular seizure, for removing, acts as a molecular seizure for removing this segment of DNA, for removing the segment of DNA, for removing or for striping, better word, this segment of DNA. So this segment of DNA, first the RNA goes and binds with the segment of DNA, and then Cas9 acts as a molecular seizure to strip it off. Clear? To strip it off. So that's CRISPR-Cas9 technology. Now we can see also that as far as CRISPR-Cas9 technology is concerned, one more thing that you have to know about CRISPR-Cas9 technology, that this technology is used for genetic alteration in reproductive cells and embryos, that you know very well. But this technology has wide application in the field of agriculture. Right now, this technology has wide application in the field of agriculture. This technology has wide application in the field of agriculture. Wide application in the field of agriculture. This technology has wide application in the field of agriculture. Application in the field of agriculture. Not only to develop hybrid varieties, not only to develop hybrid varieties, to develop hybrid varieties, not only to develop hybrid varieties, but also not only to develop hybrid varieties, but also hybrid varieties, but also but also removing diseases, and enhancing crop production, And we can see, due to its application in the field of agriculture, vast application in the field of agriculture, Due to its vast application in the field of agriculture, we can see that as far as this technology is concerned, due to its vast application in the field of agriculture, we can see that for this technology, Doudna and Carpentier were given the Nobel Prize in 2020. Right? For its vast application in the field of agriculture, for its vast application in the field of agriculture, vast application in the field of agriculture. For this technology, Doudna and Carpentier, were conferred Nobel Prize in the year 2020. Were conferred Nobel Prize in the year 2020. Nobel Prize in the year 2020. 2020. Clear? So they were conferred Nobel Prize in the year 2020. And we can see that if suppose CRISPR-Cas9 technology is used for removing diseases, then it is justified. But if it is used for fulfilling the aspiration of parents, then it is uncalled for. And we'll see what is designer baby, how the aspiration of parent is fulfilled. Right on. If CRISPR-Cas9 technology, is used for, if CRISPR-Cas9 technology is used for if CRISPR-Cas9 technology is used for removing diseases, it is justified. But if it is used for fulfilling the aspiration of parents, it is uncalled for. If it is used for fulfilling aspiration of parents, it is uncalled for. Uncalled for means it's not justified. Why should you? And if it is used yes, there is a difference between them. See, high yielding varieties of seeds can be developed by embryo transfer also. It can be developed through cross pollination also. But if we are talking about CRISPR-Cas9 technology, it would be done by gene editing. CRISPR-Cas9 technology is responsible for gene editing. So uncalled for. Now, also do remember that as far as CRISPR-Cas9 technology is concerned, CRISPR-Cas9 technology was used by scientist Jian Kui to develop twins which have innate resistance to HIV. Right? This technology was used by Chinese scientists. This technology was used by Chinese scientist Jian Kui, Chinese scientist Jian Kui, to develop twins who have innate resistance to, innate resistance means inbuilt resistance, who have innate resistance to HIV virus. And for this purpose, what he did, he was responsible for altering a gene. And this gene is called as what? CCR5. So he prepared a mutated version of that gene. Right on. For this purpose, he altered a gene. He altered a gene The gene name was CCR5. And then what he did was he altered the gene, and he by using CRISPR-Cas9 technology, he prepared an mutated version of that gene so that these girls can have innate resistance to HIV. Write down, and prepared a mutated version of that genes, and prepared a mutated version of that gene. The mutated version was called as CCR5 delta 32, referred to as CCR5 delta 32. So this was done. But in this context also, we can see it can be supported also. It can be criticized also. You can support CRISPR-Cas9 technology because it has wide application in the field of agriculture. It enhances agriculture production. And for this purpose, Doudna and Carpentier were conferred the Nobel Prize in the field of medicine. Clear? It can cure diseases also associated with reproductive cells. Jiankui developed twins who have innate resistance to HIV, so it can be supported. But it can be criticized also. Criticism on the basis of what? Criticism on the basis of that why should we develop designer babies? Why should we fulfill the aspiration of parents with the help of this technology? That's the criticism. Maybe that the girl or the boy, when he grows up, he won't be approving the features that he has, which was wished by his mother or his father only. Clear? Wished by his mother or father only. So that is the criticism. Fine. But here we'll understand that what is the meaning of designer baby. We have referred to designer baby in here that it is aspiration of parents should be not fulfilled. That's unjustified. But what is the meaning of designer baby? See, designer baby means designer baby was first of all developed in a Los Angeles lab in the year 2012. And in this case, a hormonal injection is given to the female. A hormonal injection is given to the female. Given to the female means the mother, so that she releases more than one ovum at a time. Suppose 4 ovums are released by her. So hormonal injection is given to the female. She releases release of more than one ovum. More than one ovum by her. The eggs. Clear? So that would be done. Now what is done is that these eggs would be now fertilized in the lab. So what we are doing is that we are responsible for fertilizing these eggs in the lab. So in vitro fertilization takes place. IVF. In vitro fertilization takes place. So these 4 eggs have been fertilized in the lab. And they have been fertilized in the lab by either the sperm of father or a selected donor. So that has been done. Generally, selected donor is the case. Now, after this fertilization, we can see that 4 embryos would be formed. These are the embryos which are formed. Embryos which are formed. Clear? 4 embryos would be formed. Now, these embryos which are formed now would be scanned for desired characteristic which the parents wish, that whether the genetic material in these embryos are such that the desired characteristic, the aspiration of parents can be met. Clear? So the desired characteristic or the aspiration of parents can be met, whether it is possible or not. So scan for desired traits or desired characteristic. Fine, that would be done. And we can see that not only scan for desired traits or desired characteristic. So this would be scan for desired traits or characteristic. And then what we are responsible for doing is desired characteristic. This would be scan for desired characteristic. And then we are doing what? That suppose this is an embryo which shows desired characteristic. So we would be using CRISPR-Cas9 technology for gene editing of this embryo. Gene editing of this embryo to meet the requirement of parents. Meet the requirement of parents. So that exactly those characteristics are present in this embryo as wished by the parent. As wished by the parent. This is selected. So we are using CRISPR-Cas9 technology here to enhance the traits, to enhance the characteristic so that the characteristic wished by the parent is present. Clear? Now, what would be done is that this would be now selected. This has been selected. Now, this would be transferred to the womb of surrogate mother. This is now transferred. The baby that is born is designer baby. This is how designer baby is born. So everything depicted on the board, that normal hormonal injection is given to the female so that she releases more than one ovum at a time, and all these 4 ovums are fertilized in the lab. By in vitro fertilization takes place by the sperm of either the father or a selected donor. Generally the selected donor is used. Clear? And then 4 embryos would be formed. Now these embryos 4 embryos which are formed would be now scanned for desired traits, scanned for desired characteristic, scanned for desired traits, and we can see embryos which are formed would be scanned for desired traits. And now what we would be responsible for doing, we would be selecting one embryos which has the maximum number of characteristic. Clear? And then gene editing would be conducted on that to meet the requirement of parents. Clear? Through CRISPR-Cas9 technology, now the embryo would be transferred to the womb of the surrogate mother. The baby this is born, which is called, the baby that is born would be called as designer baby. Clear? So the baby that is born is nothing but designer baby. Now this you have to know. You can take a click of all these things which has been depicted on the board. Now, fine. Now next topic that you have to cover up is called as what? It is called as GM crops, genetically modified crops. GM crops, genetically modified crops. GM crops. Fine. Now, as far as GM crops is concerned, first thing that you have to know is that GM crops would be essentially having rDNA present in it. rDNA present in it means that recombinant DNA is present. rDNA means recombinant DNA is present. And recombinant DNA means that if recombinant DNA is present, it means that genetic engineering has been conducted on these crops. Clear? So first thing is that genetic engineering has been conducted. So these are also called as transgenic crops, also referred to as transgenic crops. And the first thing is that essentially it should have rDNA. Clear? So it means that genetic engineering has been conducted. The second thing is what? That not only essentially it should have an rDNA, but also we can see that as far as GM crops is concerned, GM crops are responsible for what? They are suitable for different climatic conditions. We say that they are suitable for different climatic conditions. Climatic conditions. Different climatic conditions. Clear? GM crops are suitable for different climatic conditions. That's one thing. And the second thing is what? That as far as GM crops are concerned, they are not only suitable for different climatic conditions, but GM crops, genetically modified crops, which is also called as transgenic crops, it can be used for enhancing crop production. So we can see that the purpose is what? It can be used for enhancing crop production. It is not only used for enhancing crop production, but we can see also, just wait for 1 minute, 1 second, I'm returning back. So it is used for enhancing crop production. And also we can see that it is used for what? It is used for removing diseases associated with the crop. So diseases also would be removed with the help of GM crops. And as far as GM crops is concerned, it is also used for what? It is also used for biofortification. Biofortification means to enhance the nutrient content of crop. To enhance the nutrient content of crop. So that is called as biofortification. So if suppose the nutrient content of the crop is enhanced, it is referred to as biofortification. So for this purpose, it would be used. So all these are, but it's not necessary that all fortified crops would be GM. All fortified crops would be transgenic in nature. For example, IIT Kharagpur and also the Department of Biotechnology of India developed a variety of rice which is called as what? Which is called as fortified rice. And what they did was they took uh, broken rice. They made a flour out of it, out of the broken rice, and then they developed this variety of rice, which is called as what? Which is called as the fortified rice. How they did was broken rice was taken. It was converted into flour. Nutrients were added. Vitamins were added, and then a dough was prepared. Now this dough was passed through a machine which is called as extruder to cut it in the shape of rice grains, and this was dried and this was mixed with normal rice in the ratio of 1:100. So this is called as fortified rice. This was given to the school children in India who were suffering from anemia. So this was, but there's no transgenic mechanism. It's not transgenic in nature. It's not genetically modified. No genetic alteration has taken place. Clear? And as far as the history of GM crop is concerned, the first GM crop which was developed was called as what? It's called as genetically modified tobacco. But the first GM food crop to be cultivated, was called as what? Was called as GM tomato. So from tobacco to tomato was called as GM tomato. So first GM crop which was developed was called as GM tomato. So that's the history. But we have many examples of GM crops. Clear? Some examples of GM crop, if we talk about some examples of GM crop, we can see that the first example is of golden rice. Golden rice. And why golden rice? Now rice has a deficiency of beta carotene present in it, which gets transformed into retinol or vitamin A inside the body. So if suppose we are talking about rice, we consume only and only rice and we think that vitamin A is present in our body. Not at all, because rice is lacking vitamin A, because rice lacks beta carotene which gets converted into retinol or vitamin A inside our body. So to remove this drawback of rice, golden rice was developed by two scientists, Peter Berger and Ingo Potterkins. So they developed this variety and they took genes from other organisms for this purpose. Since the color of beta carotene is golden, hence the name golden rice. So it's not that rice only is lacking beta carotene. Rice also is lacking iron and zinc. So iron and zinc is also lacking in rice. So you can see that iron and zinc is also lacking in rice. And as far as the scientists of Italy, they developed a transgenic variety of rice to remove these drawbacks. And this variety of rice was called as what? This was called as super rice. So we can see through transgenic means the iron content has been enhanced 4 times and the zinc content has been enhanced twice. So the iron content has been enhanced 4 times and the zinc content has been enhanced twice. So this variety of rice is called as what? This variety of rice is called as super rice. Super rice. Many examples are there. But in India, a concern is what? The concern is of 3 main varieties, which BT cotton, BT brinjal and GM mustard. Let's discuss all these things. You can take a screenshot of this and then write about golden rice and super rice, because everything has been written. GM crops, transgenic enricher, consists of rDNA suitable for different climatic condition. Purpose is what? To enhance crop production, remove diseases, biofortification. First GM crop developed, GM tobacco, and first GM food crop, GM tomato. GM tomato was called as flavor saver. The name was given as flavor saver. This much you can note, and then I'll dictate the two, golden rice and super rice. Take a pic of this or write down, please. Take a pic of this part. That what is GM crop and what are the objectives of producing GM crops. Clear? What are the objectives of producing GM crops? Write down. There are numerous examples of GM crops. There are numerous examples of GM crops. This I haven't written, so I'll dictate these two. This already depicted on the board. There are numerous examples of GM crops. First, golden rice, golden rice, golden rice. Rice has a deficiency of beta carotene. Rice has a deficiency of beta carotene, which gets converted into retinol, or vitamin A, inside the body, which gets converted into retinol or vitamin A inside the body, converted into retinol or vitamin A inside the body, Clear? To remove this drawback, Peter Berger and Ingo Potterkins, to remove this drawback, Peter Berger and Ingo Potterkins developed a transgenic variety called golden rice. By taking genes from other organisms, since the color of beta carotene was golden, hence the name golden rice. Since the color of beta carotene was golden, hence the name golden rice. Second, super rice. Rice also has a deficiency of iron and zinc. Rice also has a deficiency of iron and zinc. To remove this drawback, scientists of Italy developed a transgenic variety, called super rice. developed a transgenic variety called super rice, in which the iron content iron content has been enhanced four times. The iron content has been enhanced four times, and the zinc content twice, and the zinc content twice, zinc content twice. Clear? So this variety is called as super rice. But in India, there are three much talked about varieties. One talked about variety is called as what? It is called as BT cotton. Second much talked about variety is called as BT brinjal. And third most talked about variety nowadays is called as GM mustard. So what is BT cotton, what is BT brinjal, and what is GM mustard? Now when we talk about BT cotton, cotton suffers from a disease which is called as bollworm disease. Bollworm. The balls of cotton are affected by insect. So that disease is called as bollworm disease. Now, in order to remove this drawback, a company called Monsanto of US developed a transgenic variety. And this transgenic variety is called as what? This transgenic variety is referred to as BT cotton. Why? Because the gene for this purpose has been taken from a soil bacterium. The gene for this purpose has been taken from a soil bacterium. Clear? And the soil bacterium is called as Bacillus thuringiensis. The gene for this purpose has been taken by a soil bacterium, and the soil bacterium is called as what? It is called as Bacillus thuringiensis. Bacillus thuringiensis. Clear? Thuringiensis. Fine. So gene has been developed from a soil bacterium. Now, what we can see is that as far as these genes are concerned, the genes are what? The genes are Cry1AC, Cry1AB, and also Cry9C. Now, what are these genes? These genes are called as what? They are called as protoxins. Protoxin means, see the words, protoxins, protein, and toxins. So they are essentially proteins, but they would be converting into toxin when the environment is altered. And in which environment they would be converting into toxin? They would be converting into toxin in alkaline environment. Alkaline environment. So these genes are Cry1AC, Cry1AB, Cry9C. So they would be converting into toxin when the environment is altered, when the medium is altered. And in alkaline medium, they would be getting converted into toxin. So if suppose this is the insect which is responsible for causing bollworm, and this is the gut of the insect. So when the cry gene reaches the gut of the insect, the gut of the insect is alkaline. Human gut is what? Acidic. So it won't be get activated in the acidic gut. But we can see cry gene would be getting activated in the alkaline gut of insect. And if this be the case that they are getting activated in alkaline gut of the insect, they would be alkaline gut. They are getting activated in the alkaline gut. So you can see that this cry gene activated in the alkaline gut, this cry gene is getting converted into toxin. So this would be forming numerous pores on the walls of the gut. So the gut becomes porous. Cry gene, since it is a protein, it is getting converted into toxin. Why? Because of the alkaline gut of the insect. So it would be forming numerous pores on the walls of the gut, because of which the insect dies. So this is how it is responsible for removing the problem. Clear? Because if the insect dies, the bollworm disease would be reduced. So we can see BT cotton was cultivated on a large scale in states like Maharashtra, Madhya Pradesh. And what happened was that initially the cultivation was very good, but subsequently the cultivation reduced. And the allegation was put on BT cotton that BT cotton is the variety which is like this, that one or two generation you can have good cultivation, but subsequent generation you would be having the production less, because the allegation against BT cotton was that if it is repeated cultivated, the fertility of soil would be reduced. But this should not be done. Why? Because still in India, the cotton which is 70% of the cotton which is cultivated is nothing but BT cotton. Is nothing but BT cotton. Clear? And it is a success story worldwide. Why should we be responsible for criticizing this? Clear? Maybe that the impact assessment norms related to these transgenic crops are not very developed in India, but we should not be responsible for criticizing it. Similarly, we can see that Mexico is a company, Maharashtrian company, and in India there is a huge problem in cultivation of brinjal, because brinjal itself suffers from a disease which is called as fruit and shoot borer disease. You can see insect inside the brinjals. Fruit and shoot borer disease. And due to it, what happens is, due to fruit and shoot borer disease, what happens? The production of brinjal is less, although we have many varieties of brinjal. So the production of brinjal is less due to fruit and shoot borer disease. So in order to remove this drawback, a company called not Monsanto, it's a company called Mexico developed a transgenic variety of brinjal in which the same gene, that is Cry1AC, Cry1AB, Cry9C, were taken from the same bacterium, Bacillus thuringiensis. But see, sorry, there were many allegations against BT cotton and against the cultivation of BT brinjal. There were a number of campaigns started, so it was never cultivated as such. So although the GEAC approved it, Genetic Engineering Appraisal Committee approved it, but was also responsible for imposing a ban on it. Ban on it in the year 2009 because of the pressure exerted by these campaigns. Then comes the story of GM mustard. That what is GM mustard? Clear? What is GM mustard? Now, let's write down first of all these two and then come to GM mustard. BT cotton. BT cotton. BT cotton. Cotton suffers from a disease. Cotton suffers from a disease called bollworm disease. Cotton suffers from a disease called bollworm disease. Bollworm disease. Due to which its production is less. Due to which its production is less. US company called Monsanto. US company called Monsanto. US company called Monsanto developed a variety of cotton. US company called Monsanto developed a variety of cotton, transgenic variety of cotton. Developed a transgenic variety of cotton called BT cotton. To remove this drawback. And for this purpose took genes from soil bacterium Bacillus thuringiensis, soil bacterium. Bacillus thuringiensis, thuringiensis, soil bacterium. Bacillus thuringiensis, Bacillus thuringiensis. That's why it was called as BT. The genes included, the genes included cry1AC, cry1AB, cry9C. The genes included cry1AC, cry1AB, cry9C, cry9C. And these genes, cry genes, now they are considered as what? They are considered as protoxins. Write down. These genes are considered as protoxins. These genes are considered as protoxins. Toxins. Considered as protoxins. So essentially they are what? Essentially they are proteins, but would be converting into toxin when the environment gets altered. Write down. Essentially they are proteins. Essentially they are proteins. Essentially they are proteins, but would be converting into toxins. Essentially they are proteins, but would be converting into toxins. Converting into toxins. When the environment is altered, but would be converting into toxins. When the environment is altered, but would be converting into toxins. When the environment is altered, Clear? Fine. So we can see, and it would be getting activated where? It would be getting activated in the alkaline gut of insect. Right down. It gets activated in. It gets activated in the alkaline gut of insect. See, it won't be getting activated in the acidic gut that we have. It gets activated in the alkaline gut of insect. Alkaline gut of insects. Forming numerous spores. Forming numerous spores. Numerous spores. Forming numerous spores. Due to which numerous spores on the walls of gut. Forming numerous spores on the walls of gut. On the walls of gut. Due to which the insect dies. Due to which the insect dies. The insect dies. Clear? So BT cotton was cultivated on a large scale in India, but there were allegations against BT cotton for reducing the fertility of soil. Right on. BT cotton was cultivated on a large scale. BT cotton was cultivated on a large scale. On a large scale. BT cotton was cultivated on a large scale. Oh. Cultivated on a large scale in states like Maharashtra, Gujarat, in states like Maharashtra, Gujarat, Madhya Pradesh, Maharashtra, Gujarat, Madhya Pradesh. But there were allegations against it. But there were allegations against it. Allegations against it. That it reduced the fertility of soil. There were allegations against it that it reduced the fertility of soil. And also caused rashes on the skin of farmers. And it also caused rashes on the skin of farmers. Rashes on the skin of farmers who cultivated it. Rashes on the skin of farmers who cultivated it. Who cultivated it. Clear? So that's about BT cotton. Fine. BT cotton. Now next, that is BT brinjal. Brinjal suffers from a disease called fruit and shoot borer disease. Fruit and shoot borer disease. Fruit and shoot borer disease, due to which its production is less. Due to which its production is less. Production is less. A company of Maharashtra called Mehko. A company of Maharashtra called Mehko. A company of Maharashtra called Mehko developed a transgenic variety of Mehko developed a transgenic variety of brinjal. Develop a transgenic variety of brinjal using the same genes, that is the cry genes, using the same genes from the same bacterium, that is BT, Bacillus thuringiensis, BT. And see, had it been cultivated, BT. Now, this brinjal was given approval by the GEAC in the year 2009, but soon it was banned also because of a number of campaigns against it. Write down. This variety was given approval by GEAC. This variety was given approval by GEAC, that is Genetic Engineering Appraisal Committee. Approval by GEAC, Genetic Engineering Appraisal Committee. In the year, Genetic Engineering Appraisal Committee will continue for a while. We'll complete this. After this, GM mustard would be completed, and then we'll end the class because a lot needs to be studied. Now, this variety was given approval by GEAC, that is Genetic Engineering Appraisal Committee. In the year 2009. Genetic Engineering Appraisal Committee in the year 2009. But soon a ban was imposed on it. Ban means ban on its cultivation. Ban was imposed on it by MOEF. But why a ban was imposed on it by MOEF? The ban was imposed on it by MOEF because the ban was imposed by MOEF. When you are talking about the ban was imposed by MOEF, why? This ban was imposed by MOEF, and the reason was what? The reason was that a number of campaigns were against it. Right down. As there were a number of campaigns against it. See, if it was cultivated in the year 2009, it would have become the first transgenic crop to be cultivated in India. Clear? The first transgenic crop to be cultivated in India. Fine. Now, next variety is called as what? It is called as GM mustard. So we are talking about GM mustard. mustard. Clear? And when we talk about GM mustard, remember that in mustard cultivation, there are two problems. One problem is what? One problem in mustard cultivation is that mustard is a self-pollinating crop. So hybrid varieties of mustard is not possible. Clear? Second problem in mustard cultivation is what? That when we cultivate mustard, weeds develop in between. And when weeds develop in between, so the commonly used herbicide today is what? It is called as glyphosate in India. Glyphosate is a commonly used herbicide which has been developed by Bayer company. Bayer company. So glyphosate has been developed by Bayer company, and uh, we can see that this variety, glyphosate, when it is used on the weeds, weeds die, and also the mustard die. When you are using glyphosate, the herbicide developed by Bayer company, Bayer is a German company which is used in India on a large scale. Now, when you spray herbicide on the weeds, not only weeds die, but also the mustard dies. But also the mustard dies. So we can see that for this purpose, to remove this drawback, GM mustard has been developed. And GM mustard is also called as what? It is also called as Dhara mustard hybrid 11. Dhara mustard. So that's completely wrong. It is also called as what? It's called as Dhara mustard hybrid 11, or we can see DMH 11. How it is developed. So the scientists of Delhi University, headed by Dr. Deepak Pentel, was responsible for taking up this step of developing a transgenic variety of mustard. And for this purpose, he took what? The team took first two varieties of mustard. One variety of mustard was called as what? One variety of mustard was called as Varuna. Now, Varuna is what? Varuna is an Indian variety. And second variety was called as what? Second variety is called as Hera. Hera is what? Hera is a East European variety. So one is called as Varuna and second is called as Hera. Clear? And then they took a bacterium, and this bacterium is called as what? It's called as Streptomyces. And with the help of this bacterium, they were responsible for, first of all, transferring a gene from Streptomyces to Varuna, and also a gene from Streptomyces to Hera. Clear? Now, the gene which was transferred from Streptomyces to Varuna was called as what? It is called as Barnase. And the purpose was to make it what? The purpose was to make it male sterilized. Male sterilized means, if you see the four whorls of flower, androecium is the male part. So androecium would become inactive. What would be active is gynoecium only. See, I'm just coming to it, that how we would be dealing, because all these are not available in the books. All these are from the newspaper itself. So go through Science and Technology of the Hindu newspaper, and from there you can get the questions. Fine. And also we can see that there are some sites like emerging technologies. From here also, although there are many technologies there. So what would be referred to in Hindu can be asked as questions. Can be asked as questions. So if you go through the science and technology segment of Hindu, or there's a book also, Hindu Speaks on Science, this kind of question can be answered, which I am discussing. Clear? So go through that. Every week you go through science and technology segment of Hindu. And if suppose new development takes place, that comes out in the newspaper every time. So there's no way other than the newspaper too. Because see, I have taught you now. Next year you appear in the mains examination. And in the meantime, there will be new developments. How to tackle that? How to tackle that? Clear? So that's why I'm suggesting that before the examination, you make a collection of all these things, which has happened in the last year, science and technology segment. And then some backgrounders also. For example, CRISPR-Cas9 technology, 2 or 3 years more, they would be asking the question. Then comes a new technology. Clear? Or GM mustard, they haven't asked you question, so they can ask you question on that. Like this, lab-grown meat, what is that? Moza meat, what is that? These kind of questions would be framed in the examination, would be asked to you in examination, and they are generally in the news. See, biotechnology is a part of your mains examination. But questions on biotechnology is also asked in your preliminary examination. See, bacterium is singular in nature. That's why we say bacterium. Bacteria is generic. Fine. And that's why questions are asked on biotechnology. So in your preliminary examination syllabus, general science is general science. One can understand is basic physics, chemistry, and biology. Not a single question on physics, chemistry, and biology would be asked in your examination. They would be asking you questions from biotechnology. Some questions, 1 or 2 from nanotechnology can also figure. So best way out is to prepare from newspapers. Clear? Now, varnish. And we can see the second gene is which was taken from Streptomyces. And this gene is called as, which was introduced in Hera. So this gene is called as what's called as Barstar. Barstar. And this was just for the reverse effect. Here we are saying male sterilized, means the flower of Varna, the male part would be inactive, the female part would be operative. Here just the reverse. It becomes female sterilized. Female sterilized. So you can see the female part is active, but a female part has been sterilized, but the male part is active. And some additional genes were added to Varna. And these additional genes which were added to Varna, was for the purpose of making it HT. HT means herbicide tolerant. Herbicide tolerant, that if you spray herbicide on it, it won't be getting destroyed. And then what happened was cross pollination was conducted between the two. That is Varna and Hira. The resultant was what? The resultant was DMH11. Dhara mustard hybrid 11. That was the resultant. And this is a hybrid variety. Clear? This is a hybrid variety and this is HT in nature. Herbicide tolerant in nature. Tolerant in nature. So it means that if we now cultivate DMH11, which is GM mustard, and sprinkle glufosinate on it, then the weeds would be destroyed, but not the mustard. So this is called as DMH11. Clear? DMH11. See, the traditional biotechnology is given in the NCERT 11th, 12th book. But that is hardly asked in an examination. What they asked you is what is in news related to biotechnology. Clear? So don't go through that. Go through the news items. News items. Mainly from Hindu's science and technology. They can ask you. And also as a news, suppose it is present that this lab grown meat is in the news. Fine. Or artificial blood is made. So how it is made? Let's see. This all these things. Write down. GM mustard. GM mustard. And it's called as what? Dhara mustard hybrid. Take a screenshot of this also. Dhara mustard hybrid 11. GM mustard. In mustard cultivation. In mustard cultivation. Weeds grow. In mustard cultivation, weeds grow. Weeds grow. And to remove these weeds, in mustard cultivation, weeds grow. And to remove these weeds, the commonly used herbicide, the commonly used herbicide in India is glufosinate. The commonly used herbicide in India is glufosinate. Is glufosinate. But this not only destroys the weed, but this not only destroys the weed, not only destroys the weed, but also mustard. But this not only destroys the weed, but also mustard. Also mustard. But also mustard. Clear? And the second problem is not only this is the problem, but the second problem is what? That mustard is a self-pollinating crop. Right on. Mustard is also a self-pollinating crop. Mustard is also a self-pollinating crop. If self-pollination takes place, how can cross-pollination be possible? Here we have so self-pollinating crop. Now we have genetically altered it for cross-pollination. Mustard is a self-pollinating crop. As such, hybrid varieties of mustard, is not easily possible. As such, hybrid variety of mustard is not easily possible. A scientist of Delhi University, headed by scientist of Delhi University, headed by Dr. Deepak Pentel, headed by Dr. Deepak Pentel. Deepak Pentel, scientist of Delhi University, headed by Dr. Deepak Pentel. Deepak Pentel developed a variety of mustard. Developed a transgenic variety of mustard. Developed a transgenic variety of mustard called DMH11. Just one second. Sir, this year most of the questions were not asked from any particular segment, even they asked from species related to questions most. Sir, when your analysis it in the video 2023, it is merely simple, but when I do, I am not able to connect to the point. Even I am from geography background, sir, something about most those types of question from the see as far as species question is asked. And let me share you a list. You have reminded me, otherwise I'll forget. Let me share you a list and how to tackle those questions. Just I'm sharing you one list. and I'm sharing the group itself. Just share me that, let me share that list. Clear. So I've shared one list, and see, look at this list. This list consists of what? It consists of the major national parks and sanctuaries in India. But you know national parks and sanctuary where they are located, nothing on that. But look at the species found in that. Now, species found in that, for example, suppose black bear, or suppose musk deer, hangul, or suppose cheetal. Now, these are the animals which you have to look into. That what is their characteristic. Now, hardly we can see Chausingha, or suppose Barasingha, or Great Indian Bustard. All these, if you see, whether it is Great Indian Bustard or etc., Nilgai, or even suppose the Siberian crane. Look at all these animals and try to find out some characteristic of these animals. There would be hardly 70, 80 animals like this because some are common in all national parks and wildlife sanctuaries. Clear? To find out the characteristic of these animals, where they are found, in which habitat they live, and what is their IUCN status. IUCN status. If you do so, then the questions which have been asked in examination this year would be answered. Otherwise, it's difficult to answer those questions. For example, sloth bear. So, what is the characteristic of sloth bear? Where it is present? Okay, it is present in the Nilgiris Mudumalai sanctuary. And what is the characteristic? What is the status of sloth bear? So, just I have prepared a list. Just go through this. Flamingos, different types of flamingos. What is their characteristic? So, go through this list. And they are not very, the numbers of species are not very high as such in this list. Clear? In this list. So, prepare 2, 3 lines about those animals. And if you do so, then we can see that then the questions would be solved. Otherwise, there is no way because in the books it has not been mentioned. So I saw that there are some leads that these animals are found in the national park and wildlife sanctuaries of India. So let's make a list of these animals. And then I have prepared this list for you all and just go through these animals. Search that what is sloth bear, what are the characteristic, what is the IUCN status of that. Clear? So then the questions would be answered. Otherwise, if you are having background of geography, when you won't be able to answer those questions. It's a truth what you have said. Because the nature of questions were like this. And this year they have changed the question pattern. Clear? Although a number of things that we discussed in the class were asked as questions and simple to say that. But also the difficulty is when they ask you species like this, then the questions becomes difficult. So I have prepared that list. I have just forwarded that list. Clear? Into your group and go through that list once. Go through that list once. Clear? Right now, a team of scientists of Delhi University, headed by Dr. Deepak Pentel, developed a transgenic variety of mustard. Called DMH11. Called DMH11. That is Dhara Mustard Hybrid 11. Take a click of this also. Although I'll make you write also. There were two varieties taken. Varuna, an Indian variety. And Hera, East European variety. There were two varieties taken. Varuna, an Indian variety, and Hera, an East European variety. From bacterium Streptomyces. A gene called Barnes, a gene called Barnes was transferred to Varuna. A gene called Barnes was transferred to Varuna to make it male sterilized. Transferred to Varuna to make it male sterilized. To make it male sterilized. From the same bacterium. From the same bacterium. Male sterilized means androecium won't be functioning. The male part won't be functioning. From the same bacterium. Baristar. From the same bacterium, a gene called Baristar. A gene called Baristar was introduced in Hera. Was introduced in Hera. Introduced in Hera. For just the reverse impact. Was introduced in Hera for just the reverse impact. Just the reverse impact. Just the reverse impact. Clear? So female sterilized. And then we can see additional genes were added into Varna to make it HT. HT means herbicide tolerant. Write down. Additional genes were added to Varna. Additional genes were added to Varna. Additional genes. Additional genes were added to Varna to make it herbicide tolerant. To make it herbicide tolerant. And cross pollination was conducted between the two. And cross pollination was conducted between the two. Cross pollination was conducted between the two. Coordination was conducted between the two. The resultant variety DMH11, the resultant variety DMH11 is not only hybrid, but also HT in nature. HT in nature means herbicide tolerant in nature, but also HT in nature. HT in nature. Now, if you cultivate this variety, then what will happen is that weeds would be growing. But if you sprinkle glufosinate, weeds would die but not the mustard. Clear? These are the chapters that you have to know, topics that you have to know. From here we start disaster management. Clear? And try to complete disaster management. So try to discuss all the major parts of disaster management. Fine. And we'll have a bit of extended class next day also, so that we can complete all these things. Fine. Thank you so much. In disasters, what you have to do? First of all, you have to know about what? We have to know the definition of disaster. What is the definition of disaster? Then some disaster needs to be discussed. Main disasters needs to be discussed. Clear? And then we have to deal with some initiatives also. If they are factuals, I'll provide to you and discuss all those things. Fine. So all those things would be discussed in the next class. Thank you so much.